right, people, in our unit yesterday, we did a photosynthesis lab, and we saw that the leaves that floated went through photosynthesis. So something's got to be going on in the biosphere why we're running out of CO2, which helps us create energy storage molecules. So our next question we're trying to figure out is what caused carbon dioxide to decrease in the biodome? What caused that CO2? We saw in our simulation in the plant cell, they take in water, CO2, there's a chemical reaction, and from there we create this ESM, which is not actually energy, it's just potential energy. It's just an energy storage molecule that can be used later. So the mitochondria then, we have another process. This is photosynthesis. They take in the energy storage molecule. They take in oxygen. There's a reaction where CO2 is released, water is released, and energy is released also during the process. So maybe something's going on here then with the CO2 levels also of why our plants and animals are running out of energy in there, in our ecosystem. So we're gonna look at something called yeast. And when you look at it under the microscope, it is also a living thing. It's made of cells. We can look at this later, maybe next week. Those cells, they need food energy too, and they also go through cellular respiration like us. So with our lab today, we have our test tube racks, our yeast. Here is our yeast, and all we're gonna do is we can do some tests now. We know that it needs energy, sugar, and it needs air. So we have a balloon here also for that to close off air. So the first step, let's make it easy. All we're gonna do is take a teaspoon, the bigger spoon, there's 2 spoons. Let's take the bigger spoon, we're gonna go with the teaspoon. We're gonna teaspoon in 1 into each one. Notice each one, just putting a tablespoon in. So now there is yeast in each one. Now, in order to get the reaction going, what yeast really likes is they like water. So you're gonna have a graduated cylinder. We're gonna fill it up. We're gonna have 10 milliliters, no, all the way up top. We're just gonna put in 10 milliliters of water in each one. I already have the other ones filled up. You're just gonna have 1. You can fill it up each time. And then, and finally, 4. So they all have water, so we can get a reaction going. Now we saw the first one, they need sugar. So all we're gonna do then is we're gonna use the littler, the smaller one. We're gonna put 1 teaspoon of energy. So they have the sugar and they have the air coming from the top. So hopefully this yeast can start start going through the energy it needs. Notice that already there's a little bit of reaction going on here. There's some bubbles going on. What if we didn't give them energy storage molecules? Is cellular respiration gonna happen? So we're gonna go no sugar on this one. But then what about air? Do they actually need air? This one, we just see that it has sugar in here. So I'm gonna give this one sugar. But I say, let's close it off. Let's put a balloon on here and let's close it off from the air. So all you're gonna do is just cap it off. See, it's gonna have, no sugar. Or sorry, sugar with no air. Then our last one, if this one has no air, what if we cut off air and no sugar? Let's just put a cap on that. How do we measure cellular respiration? How is this gonna affect? Well, you have a ruler, and let's just measure in centimeters. We'll time for about 10 minutes, and we'll measure the foam height afterwards to see how much cellular respiration happened. So we'll measure in centimeters, the smaller side here for cellular respiration. That is it. Good luck, everyone.